Hello, everyone. Good afternoon. Go ahead and type your first name or your nickname into the chat for me, if you would. Thank you. Thank you very much for doing that. It's good to see everybody. Such a beautiful day today. So exciting that the weather's getting warmer and spring is in the air. I hope everybody's doing well. Today, we're going to be talking about oxygen. We're going to be talking about categories that we can put microbes into based upon their oxygen needs. So before I get started, let me ask if anybody has any questions about anything that they've been reading or watching in lecture, questions about anything they've seen on a quiz, or if you think of a question, feel free to type it into the chat. We can always talk about things like that when we get together for lab. I think what I'll do first is I'm just going to pull up our schedule. I'm going to pull up our Canvas page so we can see what's coming up. So this is not Canvas. There we go. So you should be seeing our Canvas page that has the modules on it. This is what the page looks like up at the top, and I'm just scrolling down to this week, which is the week of March 22nd. And if you click on the to-do list this week, which I'm sure you've done, you've got the two lecture topics this week's microbial growth and control of microbial growth. And of course, both of those have a lecture quiz associated with them. And we have two different topics for lab this week as well. We have oxygen requirements today and control of growth with chemicals, with chemical disinfectants on Thursday. So we will be meeting twice this week. There's a lab homework assignment for you to complete and get into me by Sunday at midnight. And this is the week that your pathogen assignment begins. So if we go back to where we were a moment ago, on the modules page, you can see there's a place that is labeled as pathogen project. There's an instruction sheet here at this link. And then there's another link for you to submit your assignment when it's finished. This is a 50-point assignment, so this is worth putting some time and effort into. I'm sure you put time and effort into all your work, but this is one that is worth a nice big chunk of points. I'm just going to click on where it says the assignment, and just scroll through this just a little bit. I'm going to let you read through this on your own, but basically what you're going to do for this assignment is you're going to choose a set of questions to answer about a particular pathogen. So question group 1 is all about the pathogen that causes tuberculosis. And you can see there's 6 numbered questions here. And then the second group of questions right here is about the organism that causes cholera. And there's a series of questions here. Then there's a series of questions about a virus, the Zika virus. And lastly, there's a series of questions about Staphylococcus aureus. So you get to choose which organism you want to do, which set of questions you want to answer for this assignment. And you're going to be answering each of the questions by writing your answers in paragraph form. So it is a writing exercise as well as an exercise in doing a little research about these pathogens. A couple of key things to note about the assignment. For each question, each of the numbered questions that you're answering, you're going to need to use at least 3 different source materials. And after you answer each question, you're going to list your sources. So you're not going to list all your sources at the end of the assignment, but you're going to list them under each question. Now you can use a source multiple times. You might use the same source for each question. That's fine. That's not a problem. You just want to let me know that. And when you start looking for information, I've given you a list of good places to start. You can certainly use other websites. You can use your textbook. You can use any other source material that you find helpful. But I've given you a nice list here of some reputable, well-maintained websites that you're welcome to use as you do your research. The key thing is that you're going to want to let me know underneath each question what 3 or more sources that you use to answer that question, so I can go back in and look at what you used. You're welcome to talk about this assignment with each other. I have set up a discussion board for this. Obviously, there are 4 sets of questions, so it's likely that whatever set of questions you choose to answer, there'll be somebody else in class who is also working on those questions. And you can communicate with each other over the discussion board if you'd like. And you can share whatever information you're finding, or you can talk about information that you're finding. You can post questions about it if you're having trouble with one of the questions. And you can talk to each other and share what you've learned. The only thing to remember is that everybody needs to hand in their own assignment at the end of this, written in your own words. But you are absolutely free to work together on this if you find that helpful. And again, just feel free if you want to post on that discussion board and say, hey, I'm going to do the cholera questions. Is anybody else working on the cholera questions? And you can, connect with each other that way. So the instruction sheet is pretty complete, but take a look at it. And if you have any questions about what you're supposed to be doing, feel free to message me or bring your questions to lab someday. That's fine. There's some other information in the assignment sheet that you'll find when you read through it. This is both an assignment that is designed to get you to read a little bit about a pathogen and learn about a pathogen, but also to practice your writing. We always all need practice. Doesn't matter what you do in life or how old you get, everybody needs to always be practicing their writing because it is such a difficult skill to master. So, so take a look at that when you have a moment. And again, let me know if you have any questions about it. I'm going to go back now and pull up our Canvas screen again. I just want us to take a look at what's coming up. If we go forward to next week, just so you have this on your radar, we have a very unusual week next week. So that's why I wanted to call your attention to it. You actually have 2 exams in 1 week and no laboratory meetings next week. So just so you know, this is coming up next week. You have a practical exam, your second practical exam on Tuesday next week. And just like the first one, you'll want to take it sometime between 8 a.m. and 8 p.m. You want to get it submitted by 8 p.m. It's going to look very similar to the first lecture exam, the first lab practical exam. It's just covering all of the new lab topics that we've discussed since the first lab practical. So microbes in the environment, special media, the oxygen requirements material we're going to do today, and controlling growth of microbes with chemicals, which is what we'll be doing on Thursday. And then on Thursday next week, you're going to be taking your third lecture exam. The reason, or I should say, because you have these two exams in 1 week, you're going to notice that there are no lecture quizzes next week. There is no lab meeting next week, no lab meetings next week, and no lab homework. So I really want you focused on those two exams. There is one lecture topic that you'll need to view about antimicrobial drugs, and you'll need to do that before the lecture exam. All right. So in other words, antimicrobial drugs is going to be on the lecture exam on Thursday. So there's a little note right here. Be sure to view that antimicrobial lecture before Thursday. So the third lecture exam is going to cover all of these things, everything since the last lecture exam: biochemistry, microbial growth, controlling microbial growth, and again, the antimicrobials material. The lecture exam is going to look very similar to the first two lecture exams in terms of the types of questions that are on it and how long it is and so on. So nothing new there. You'll have the ability to take it anytime between 8 a.m. and 8 p.m. on Thursday. You just want to get it to me by 8 p.m. I did want to also just point out that there's a mistake in our syllabus that I just caught. In the syllabus, it lists both exams for Tuesday, March 30th, but that's not how it's done. On Tuesday, you'll do the lab practical exam, and on Thursday, you'll do the lecture exam. So if you happen to see that in the syllabus, don't panic. You're not taking two exams on the same day. So that's just to keep that on your radar for next week. It's an unusual week. It's the only week that it happens in this course that you have two exams in 1 week. And of course, you are allowed to have your lab notes next to you when you take the lab practical exam, and your lecture notes with you when you take the lecture exam, just like you did in previous exams. So does anybody have any questions about our schedule? About anything that's due this week or what's coming up next week? Any questions at all? Feel free to type them into chat. So what we're going to be talking about today, again, has to do with oxygen. We're going to talk about how we can evaluate an organism's relationship to oxygen, if you will. That's probably the best way to put it. We know that different microbes have different needs for oxygen. Some of them find oxygen actually toxic. Some of them can tolerate oxygen, but they don't use it. We see all kinds of different evolutionary adaptations regarding oxygen in microbes. Of course, there are some microbes, some bacteria particularly, who require oxygen, just like you and I do. They have to have oxygen available to them or they won't survive. So here's a question: why? Why? Why do we need oxygen? What do we do with oxygen? What do our cells do with oxygen? You and I have to have oxygen at all times, or we will quickly die. If you know what your cells do with the oxygen molecule, please type it into chat for me. So you know you have to have it. I mean, if a person stops breathing, they're on death's door, right? If a person has a heart attack, for example, and stops breathing, they're close to death, right? When the ambulance arrives and the paramedics or the EMTs get out, and the very first thing they're going to do is check the airway, right? Get an oxygen mask on them. Why? What is it that's so vital about oxygen? Why do our cells need it? Do you have any sense of that? oxygen is a critically important molecule to us. It's interesting though, oxygen is also a bit of a toxic molecule to us. How much oxygen is in the air, by the way, percentage-wise? I think sometimes people have the general public has this idea that the air is just oxygen, but of course that's not true. Does anybody know what the percent of oxygen is in the air that we breathe? Any sense of that? Type it into the chat if you do. There's actually not all that much oxygen in our air, is there? And yet it's so vital to our survival, so vital to our health. Nobody knows how much oxygen is in the air? It's right around 20, 21%. That's how much oxygen is in room air. Most of the rest of it is nitrogen, believe it or not. But yeah, about 21%. And we breathe it in constantly because our cells have an ongoing need for it. And the reason our cells have a need for it is because our cells must continually manufacture a molecule called ATP. They must continually produce ATP in order to survive. All cells on this planet need an ongoing source of ATP, or they can't survive. All cells use ATP as a source of energy in order to do the work that cells do every day. And unfortunately, cells do not store large amounts of ATP. They make it as they need it. How long does a person have when they stop breathing? How long before a person starts dying, starts losing brain cells, and is close to death when they stop breathing? How many minutes do they have? And again, I always use the example of a person having a heart attack. If a person has a heart attack and falls to the ground unconscious and stops breathing, that's an emergency, right? That's a life-threatening situation. How many minutes before their body gives in and dies? Any sense? It's a good guess, Izzy. It's typically a little bit longer than that. Izzy's guessing 3 minutes. It's typically a little bit longer, but not by much. It actually varies, believe it or not, according to what the person was doing when they had the heart attack. So if they were sedentary, like sitting on the couch when they had the heart attack, they actually may have a couple more minutes. But if they were running a marathon or something, they actually probably have fewer. So 4 to 6 minutes is the range that you'll read in most textbooks before brain cells start dying. Not much ATP. That's all you have. You have a few minutes worth of ATP. And if you don't have any oxygen available to you because you've stopped breathing, you stop making ATP in your cells and you just use whatever's left until it runs out. And then you die because you cannot survive without it. Your cells cannot survive without ATP. Now, you and I are aerobic. We do aerobic cellular respiration, which is a long series of reactions that take a carbohydrate called glucose and break it apart, break it down bit by bit by bit, break covalent bonds in the glucose molecule. And remember, all a covalent bond is a pair of electrons, right? That are being shared by two atoms. That's all it is. So when those bonds in glucose get broken, those electrons get released. The glucose is going to be broken down until there's nothing left of it but carbon dioxide. And all those electrons that were in all those bonds in the glucose molecule, they're going to be used by the cell to build ATP. The energy that is in those electrons is going to be harvested and used to build ATP. It's a very efficient process. We're very good at it. Our cells are very good at cellular respiration. There are lots of microbes, lots of bacteria particularly, that use aerobic cellular respiration, just like we did, or we do. And what the oxygen is doing in that process is it's waiting for all of those electrons to have their energy harvested so that it can then take them on. It can accept them. Remember, cellular respiration is a kind of a process that we refer to as an oxidation reduction reaction. It's actually a long series of oxidation reduction reactions, or what we often call redox reactions. And in a redox type of reaction, something gets oxidized, something gets reduced. Something loses electrons, that's oxidation, and then something else gains those electrons, that's reduction. Those two things must occur together. If some molecule loses electrons, some other molecule must gain them. In cellular respiration, as glucose is broken down, it's losing all of its electrons. And like I said, there's a series of reactions here going on. So it's an oxidation and then a reduction and then another oxidation and another reduction, and it goes down, down, down the line until it gets to this structure called the electron transport chain. And this chain of proteins takes all of those electrons and harvests their energy. It doesn't use the electrons themselves to make ATP. It uses the energy from the electrons to make ATP. And at the end of the electron transport chain, some final molecule has to be reduced. At the end of all of the reactions, something has to accept or gain those electrons. The glucose was the original molecule that got oxidized. Oxygen is the final molecule that gets reduced. And when oxygen receives those electrons, it actually also takes a little hydrogen and it makes water. That's why you need oxygen. You need to breathe oxygen every minute of every day because oxygen is the final electron acceptor molecule at the very end of the electron transport chain in cellular respiration. If you don't have oxygen, there is nowhere for those electrons to go, and the whole process will stop. Remember, the energy from those electrons gets used to make ATP. So if the process stops, you stop making ATP. And then you've got a couple of minutes left of ATP in the cell. So that's why you need oxygen. And that's why some microbes need oxygen. So that the oxygen can be the final electron acceptor in cellular respiration. Now, not all bacteria need oxygen. Not all of them. Some of them do, but not all of them. There are some bacteria that find oxygen toxic. There are others that don't find it toxic, but they don't use it. Some bacteria can do cellular respiration using a different molecule as their final electron acceptor. Those microbes, those bacteria are called anaerobes. They are doing anaerobic cellular respiration. They're still doing cellular respiration. They're still breaking down the glucose, taking all the electrons, sending them down the electron transport chain, harvesting all that energy. And at the bottom of the electron transport chain, there has to be a molecule that can accept all of the electrons, that can get reduced. The glucose gets oxidized, and something at the end of the electron transport chain has to get reduced. And in those bacteria, it's not oxygen. It can be several different molecules, actually, depending on the species. Some bacteria use a molecule called nitrate. Some use a molecule called sulfate. Some use carbonate. Note that all of those have oxygen in them, but they're not oxygen gas. So they're still doing respiration, cellular respiration. They're just using a different final electron acceptor. And of course, there's a third way that microbes can make ATP that has nothing to do with cellular respiration, and that's fermentation. Many microbes can do a couple of different pathways. There are even microbes that can do all three of those. They can do aerobic cellular respiration, they can do anaerobic cellular respiration, and when the need comes, they can do fermentation. All three of those things will make ATP. So, one of the things that we need to do in the laboratory often when we're evaluating a microbe, we've got a microbe, we don't know what it is, and we need to identify it. One of the things we can do is test its oxygen requirements, because that's a characteristic of different microbes. It's one of the features that each microbe has. Some of them are oxygen users, some of them don't use oxygen at all. So that's what we're going to talk about. We're going to talk about a test that we can use to determine what category of oxygen requirement different microbes have. So, a lot of information there. You'll hear me talk about respiration again and again as we move forward this semester, because it's such an important metabolic reaction for us to understand in microbiology, certainly, but also just in human anatomy and physiology. It's an important thing to understand about how we make ATP, because ATP is really the molecule of life. All living things require ATP. They can't survive without it. All right, I'm going to go ahead and jump into our lab slides. Okay, so our objectives for the exercise today are first to just talk about the 5 different categories of oxygen requirements, if you will. And I've got them listed at the top of this slide. We've got obligate aerobes, obligate anaerobes, aerotolerant anaerobes, micro aerophiles, and facultative anaerobes. So all bacteria can be put into one of those categories regarding how they relate to oxygen. Now, another objective today is to talk about a certain kind of culture medium that we use to determine the oxygen category for a microbe. And this medium is called Thioglycolate medium. We're going to talk about methods that we have available to us to culture anaerobes in the laboratory. We're going to talk about another test called the Catalase Test and why, what it does, how it's run, and what catalase is. And then we'll do a little experiment and we'll try to identify which category certain bacterial species belong to. So on this slide, I've just summarized some of the things that I just talked about with you regarding this important molecule called ATP. This is the structure of ATP up at the top. Adenosine triphosphate is what ATP stands for. And the triphosphate is right here. 3 phosphate groups that are bound to each other. And what the cell can do is break off the outer phosphate group and release a little bundle of energy when the cells require energy to do chemical reactions. As I said, there are lots of ways to make ATP: aerobic cellular respiration, anaerobic cellular respiration, and fermentation. 3 ways to do it. And depending on the species of bacteria, we see all kinds of combinations of these 3 pathways. We can test microbes to find out what their oxygen requirements are, and then basically estimate or guess at which category they belong in, what pathways they're using. Once we know what the oxygen category is for a microbe, we can better culture it in the laboratory. And of course, we can also help identify an unknown bacterium by finding out what oxygen category it belongs to. So let's talk about these 5 categories for a minute. Obligate aerobes are the category that humans belong to. An obligate aerobe is an organism that requires oxygen, again, in order to make ATP. Oxygen serves as the final electron acceptor at the end of the electron transport chain during cellular respiration. Now, obligate aerobes use aerobic cellular respiration as their primary way to make ATP, but many of them can also use fermentation. The difference here is that one of these pathways is considered the primary pathway and the other is considered a secondary pathway. In other words, if oxygen is available, this organism is going to do aerobic cellular respiration. If oxygen is not available, it's going to switch to a secondary pathway of fermentation. And because they are obligate aerobes, they can only ferment for a little while before they'll die. You and I can also ferment, right? The thing is, fermentation, while it does make ATP, it doesn't make a lot of ATP. It makes low levels of ATP. It's very inefficient. Cellular respiration makes lots of ATP very efficiently. So human beings, we don't tend to think of as being fermenters because our ATP requirements are so high. Fermentation is not going to extend our life very much when we stop breathing oxygen. But the times that you do see humans fermenting is when we tend to get into situations where we're not getting enough oxygen. We're still getting it, but we're not getting enough of it to meet our needs. Think about the person who is overexerting themselves athletically. My favorite example is the person who goes out on a weekend and tries to do exercise that they're not capable of doing. Somebody who goes out and tries to run 5 miles when, you know, they haven't run a single mile all winter. That's a person who's going to very quickly find themselves gasping for breath, right? You've seen these folks. You've seen them on the sidewalks on the weekend. This is a person who is exerting themselves past what they can possibly breathe in. In other words, they're making ATP so fast that they can't get enough oxygen by simply breathing. Now, that person's not going to die because they are still getting oxygen. So they're doing cellular respiration. They're making ATP. They're just not making enough ATP for those extended requirements of running. So what'll happen is they'll also start to ferment. And human beings use the lactic acid fermentation pathway. And in their muscle cells, they're going to be fermenting to get a little more ATP produced. And lactic acid will end up as the byproduct. People who are very, very ill, very, very ill. Think about a person who's in the ICU with COVID, for example. Those people are getting oxygen, even the folks, you know, the most sick people who are on a respirator, for example, on a ventilator, excuse me. They're getting oxygen, but they're not getting enough. Those people are going to ferment as well. So we do ferment. We just don't ferment like a microbe ferments. Microbes are single-celled organisms, so their requirements for ATP are not nearly as high as ours are. And so many of them can ferment and get enough ATP without doing respiration at all. So we've got 3 possibilities for our bacteria, 3 possible metabolic pathways. And obligate aerobes will do aerobic cellular respiration first. They require oxygen because, again, it's that final electron acceptor. All right. The other thing I have listed for the obligate aerobes is this. They also tend to have an enzyme called catalase. And this is that test we're going to look at today. The catalase enzyme is an enzyme that breaks down a toxic molecule called hydrogen peroxide. Hydrogen peroxide is a molecule that comes from oxygen. So because an obligate aerobe is taking in oxygen all the time to make ATP, there's lots of oxygen around in the cell, and sometimes that oxygen gets converted into toxic byproducts. Sometimes those oxygen molecules become what are called free radicals. You've probably heard that name. Free radicals are an important part of inflammation and things like aging of cells. But another toxic oxygen byproduct is hydrogen peroxide. And hydrogen peroxide is toxic to living cells. So it needs to be broken down. So obligate aerobes have catalase. We expect them to have a positive catalase test. Now, on the other end of the spectrum, really, the opposite category is an obligate anaerobe. These are organisms that not only don't use oxygen to make ATP, but they find oxygen toxic. Oxygen will kill this kind of an organism. They're obligate. They must be in an environment that is free of oxygen, or they'll die. These organisms perform anaerobic cellular respiration as their primary pathway to make ATP. They can also sometimes use fermentation. Again, that varies by species. These organisms tend to lack catalase. And if you think about it, that makes sense. They never have catalase and they never have oxygen inside of them. There's never going to be oxygen in that cell because if there was, the cell would die. So if you're never exposed to oxygen in your natural environment, you're never going to have toxic oxygen byproducts floating around like hydrogen peroxide. So you don't need an enzyme to break it down. So we would expect those organisms to have a negative catalase test. All right, third on the list are a group called Aerotolerant Anaerobes. Again, these are organisms that cannot use oxygen to make ATP, but they tolerate oxygen. Oxygen is toxic to an obligate anaerobe, but an aerotolerant anaerobe doesn't mind it. It's not going to use the oxygen to make its ATP, but it's going to tolerate it. It's not going to die if it is surrounded by oxygen. Now, these organisms also lack catalase. They don't have catalase, but it's not because they're not exposed to oxygen. They do get exposed to oxygen. They live in environments where oxygen is present. It's just that they literally don't have the pathway that generates hydrogen peroxide. So they just don't need any catalase. Now, here's an interesting fact about aerotolerants. They typically use fermentation to make their ATP. So these are organisms that tend to ferment. They grow best in low oxygen concentrations. Make a note of this. Put a little star next to this in your notes. It turns out that aerotolerant anaerobes actually grow best in environments where there's a low concentration of oxygen, so less than what you would find in room air. Now, take a look at this slide. This slide shows this type of culture medium that I mentioned at the beginning of class today called Thioglycolate broth. The name is right down here at the bottom, Thioglycolate. Now, it's technically not a broth, and I'll explain that in a couple minutes, but we refer to it as a broth because it's certainly not a solid. I want you to look at the different tubes in the drawing. Each tube is a tube full of this culture medium. It's been inoculated with an organism that falls into one of the 5 categories, the oxygen categories. And what the little red dots represent are bacterial cells growing in this tube. In thioglycolate medium, we're going to see what we call patterns of growth. This tube of thioglycolate, it actually has a little bit of agar added to it. Not a lot, but a little bit. And the agar turns it from a liquid broth into something that's just a little bit thicker. Now, why would we put agar into this broth tube? Well, we want to be able to create a gradient of oxygen in here. Now, because the tube is open, oxygen is going to interact with this broth, this culture medium. And you're going to have an area up at the top of this tube that has lots of oxygen in it because oxygen will dissolve right into the culture medium. But then at the other end of the tube, way down at the bottom, there won't be any oxygen because it won't be able to penetrate all the way down to the bottom. So lots of oxygen up at the top of the tube, no oxygen down at the bottom of the tube, and everything in between. So in other words, lots of oxygen, and then as you go down, there's less and less and less until there's none. So what a thioglycolate tube does is it gives us an opportunity to see where an organism likes to grow. Does it like to grow where there's lots of oxygen, like an obligate aerobe? It's going to grow right up at the top of the tube. We give this region, it's about the top 20% of the tube. We call it the oxic zone. The word is right here on the slide, O-X-I-C, oxic zone, because there's going to be plenty of oxygen in the medium right here. Way down at the bottom of the tube, that is the anoxic zone. There's no oxygen down here. Everywhere else in the tube, we also refer to as being anoxic, even though there's a little bit of oxygen here. So lots of oxygen at the top 20%, then less and less and less until there's none. Now, if you're an obligate aerobe and you are sitting in this tube, you're going to want to stay where the oxygen is. So they will grow, these cells, these red cells, will only grow in the oxic zone. If you're an obligate anaerobe, you don't want to be near the oxygen. Oxygen's toxic to you. So they grow way down at the bottom of the tube. And if we go all the way over here to this one, this is what's called an Aerotolerant Anaerobe. All right, so this is the one that we said grows best in a low concentration of oxygen. Now, you'll notice I put this green square here. Let me explain this. The way the artist drew this tube is not really accurate. Aerotolerant anaerobes do not grow evenly throughout a thioglycolate tube. See how the artist has drawn it that way? It looks like the red balls are present all the way up and down the tube evenly. In reality, you're not going to see any growth in the top half of the oxic zone, but you will see growth in the lower half of the oxic zone. Let me say that again because that's important. I've written it right down here for you. These organisms do not grow in the top half of the oxic zone, but they do grow in the bottom half of the oxic zone. In fact, they grow best in the bottom half of the oxic zone. They can also grow in the rest of the tube because remember, they don't need oxygen. They don't use oxygen to make ATP. They just happen to grow best when there's a little oxygen around. Remember, there are other things that a microbe might be doing with oxygen other than making ATP with it. So they don't use it to make ATP, but they tolerate it. And in fact, they grow best when there's just a little bit of oxygen around. But they can also grow a little bit less well when there's no oxygen around. So quite adaptive little organism, an aerotolerant. All right, let's look at the other two categories for a minute. Number 4 is called a microaerophile. This is an organism that does require oxygen to make ATP, but it can only have it at lower levels, at reduced levels. So like an obligate aerobe, it uses oxygen to make ATP. It has to have oxygen. It requires oxygen, but it can't have it at the full 21%. It needs lower levels of oxygen in order to grow. Microaerophiles grow best in low oxygen and increased amounts of carbon dioxide. They like more carbon dioxide than is in room air. So if you want to grow a microaerophile in the laboratory, it's a little hard. You've got to reduce the oxygen that they're getting exposed to, and you've got to increase the carbon dioxide. It's not easy. There is a device we use, though, called a CO2 jar. Now, what a CO2 jar has is a very tightly fitting lid. And what you do is you put your agar plate with cells on it into this jar, and then you put this special material in it that generates carbon dioxide. What it does is it sort of adds the extra carbon dioxide while forcing some of the oxygen out. In the old days, before we had this available to us, people used to just take this jar and put a lighted candle in it and then close it. So they would put the plate in, they'd put a lighted candle in, and then they'd shut the jar. And what's going to happen? Well, the candle is going to burn, right? The candle's going to burn and use up the oxygen in the jar and generate carbon dioxide. And then the candle's going to go out. The thing is, in a CO2 jar, you're not removing the oxygen. You're reducing it, but you're not removing it completely. So as long as you add a little extra CO2, you've created a really nice environment for micro aerophiles. They're a little tough to grow, though, I'll tell you. All right, the last oxygen category is the facultative anaerobe. I've got an asterisk here because most bacterial species fall in this category. Most bacterial species are facultative anaerobes. This is the most adaptive group. They are able to survive in the most varied environments. So let's talk about what their requirements are. These organisms grow best with oxygen, but they don't require it to make ATP. They are able to do aerobic cellular respiration. That's when they grow the best. They're able to do anaerobic cellular respiration, and they're able to do fermentation when oxygen's not available. Facultative anaerobes, they are the most adaptive type of organism. And if you think about it, if you're a bacterial species, especially if you're living out in the world, in the environment, this is a good way to be, right? Because there are going to be times when, you're in the air, you're exposed to the air, so you can do your preferred pathway of aerobic cellular respiration. But, you get moved around. Bacteria can't walk away. So, if there's a landslide or something, or if, an earthquake and you get sent down into a crevice, if there's a flood and you're suddenly covered in water, they find themselves in different environments all the time. So it's a very handy thing for them to be able to switch metabolic pathways according to which environment that they're in. So most bacteria actually fall in this category. They grow best when oxygen's around. So their primary pathway is aerobic cellular respiration. But no oxygen around, no problem. They'll just switch. If there's another final electron acceptor available, they'll do anaerobic cellular respiration. And if there is no final electron acceptor, well, they'll switch to fermentation. Very, very adaptive organisms. All right, so if we go back to our tubes, this is how a facultative anaerobe grows in thioglycolate. The best growth, the most growth is going to happen in the oxic zone. That's what you're seeing right here. They can also grow in the rest of the tube, but the best growth is going to be all the way through the oxic zone. So I want you to take a minute and compare a facultative anaerobe with an aerotolerant anaerobe. I've added in some colored squares here to try to fix the drawing. Remember, an aerotolerant also grows best up at the top, but only the lower half of the oxic zone. Facultatives grow all the way through the oxic zone. Both of them can grow down the rest of the tube as well. So this is the difference between an aerotolerant and a facultative. Now, the last one was the micro aerophile, and this is what their tubes look like. Remember, they require oxygen, so you're not going to see them growing down in the bottom parts of the tube, but they don't like full oxygen. They don't like room air oxygen. So they're not going to grow in the top half of the oxic zone. They're going to grow in the bottom half of the oxic zone, like the aerotolerants do. But they can't grow down in the rest of the tube because there's not enough oxygen in there. All right. Before we look at some experimental results in thioglycolate tubes, let's talk for just another minute about this Catalase test. The Catalase test is one of the most just rewarding tests to do in the micro lab because it's very easy to do and it's very fast. What this test is looking for is the presence of the catalase enzyme. Okay, some bacteria will have it, some won't. It's one of the identifying characteristics of different species of bacteria. Catalase, as I said a minute ago, breaks down hydrogen peroxide. So hydrogen peroxide in the presence of catalase is going to become water and oxygen. So such an easy test to do. You take a glass microscope slide, you pick up some culture material. You can either pick up a couple of colonies off of an agar plate, or you can get a loop full of inoculated culture broth, and you just put it onto the glass slide. Then you put a drop of hydrogen peroxide on top of it, and you watch, and it happens very quickly. If the cells that you've put on that slide have catalase, they're going to start breaking down the hydrogen peroxide, and they're going to start liberating oxygen gas. And the oxygen gas looks like this. It bubbles. So a positive catalase reaction is seeing bubbles. A negative reaction is this. Nothing happens. You put the hydrogen peroxide down on the cells, nothing happens. Have you ever put hydrogen peroxide on a wound? Have you ever put some hydrogen peroxide onto a cut because you wanted to clean it out? What happens when you put it on the cut? It bubbles up, right? It bubbles up. Your cells have catalase in them. You're an oxygen breather. You use oxygen. Your cells are going to get exposed to toxic oxygen byproducts like hydrogen peroxide. So they better have a way to break down hydrogen peroxide, and they use catalase. Now, here's a question. You're a eukaryote, not a prokaryote. You have organelles in your cells. In which organelle is the catalase enzyme found? Which one of your organelles breaks down hydrogen peroxide? What do you think? Can you remember? Can you remember? You have a special organelle whose one of their jobs is to break down hydrogen peroxide. Do you remember what it's called? It's the peroxisome. You have an organelle called a peroxisome. And the job of that organelle, one of the jobs, is to break down hydrogen peroxide. So that's where the catalase enzyme is stored inside of your peroxisomes. All right. One other thing to mention is I said it was hard for us to grow micro aerophiles in the lab. It's also really hard, really challenging to grow anaerobes. And I'm specifically talking about obligate anaerobes. It's not impossible, but you need some specialized equipment. Now, we already talked about the thioglycolate broth, and we can use thioglycolate broth to grow anaerobes. Remember, we said that broth has a little bit of agar in it. And what the agar does is it reduces the ability of oxygen to penetrate the broth. It will penetrate up at the top, but not all the way down to the bottom. Now, thioglycolate also has a chemical in it called sodium thioglycolate. And what that chemical can do is combine with oxygen. So if any oxygen gets down into the deep regions of the tube, the thioglycolate will remove it. I do want to mention that you can buy thioglycolate broth that has an indicator dye in it, and that dye will turn pink in the presence of oxygen. So if you buy that kind of thioglycolate for your laboratory, the oxic zone will be really easy to see because it will turn pink. So thioglycolate is considered a reducing medium because it combines with oxygen, gets it out of those deeper regions. There is another device that is commonly used in the lab to grow anaerobes, and it's called a Brewers jar. Like the name suggests, it's just a jar. It's a plastic jar that you can see through. It's got a very tight fitting lid. So similar to that CO2 jar. But remember, we've got to get all of the oxygen out of this jar, or it's going to kill our anaerobes. So you put in a different kind of packet. This one is not a CO2 generating packet. This is a hydrogen gas packet. You put your agar plate in, you put this gas pack in, and you close the lid. What the hydrogen is going to do is combine with any oxygen that's in the jar and turn it into water. In fact, you'll know that your hydrogen gas pack is working because you'll start to see water droplets appear on the walls of the tube. You then take your Brewer's jar and you put it right in the incubator. You put the whole jar in the incubator. And what you've done is you've created a little environment that has no oxygen in it. And in the warmth of the incubator, those cells should be able to grow. There is one other device you should just be familiar with anyway, and that's this over here in the picture. This is called a glove box. You can buy glove boxes and create a larger environment without any oxygen in it. You can see there's a little adapter here. You attach some tubing to this and you can remove oxygen from this box. It's a glove box, so you can put your hands here and into these gloves that are attached, and you can work inside the box. You can move plates around and culture tubes and so on. These are not very common unless a laboratory works with anaerobic organisms a lot. They don't tend to have these around. There are also incubators that are for anaerobic culture. So you can buy an incubator that will remove all of the oxygen from inside, but those, as you might imagine, are very expensive. And I've never seen one. You would have to have the need to make such a big purchase. You'd have to be a lab that cultured a lot of anaerobes in order to make that worthwhile. So this is what a thioglycolate tube looks like. Most of the time people buy these instead of making them themselves. It's just a little tricky to make them correctly, so we often purchase them. This is thioglycolate that has that indicator dye in it. You don't have to buy the indicator dye, but if you do, what'll happen is the oxygen will turn pink. So when you put some cells in here and you let them incubate and grow, you'll be able to tell exactly if they're growing in the oxygen or outside of the oxygen. You'll just have a more clear representation. And this is what a brewer's jar looks like. Just a plastic jar with a very tight fitting lid. The silver packet that you see in here, that's one of the hydrogen packets that comes with the jar. You can also buy what are these indicator packets. These will change color if any oxygen accidentally gets into the jar during incubation. So you'll know when you come in the next day if your jar accidentally got oxygen into it, because this white packet will turn a color. So this is what thioglycolate broth looks like when you purchase it. This does not have the indicator dye in it. That's why it doesn't look pink anywhere. But remember, you really don't need the indicator dye because the top 20% of the tube is where the oxygen is. And I think you can see if I tilt this, there's no microbes in here. This is just uninoculated broth. But if I tilt it, I think you can see it's just a little bit thick. See that? It's just a little bit thicker than a broth would be. And that agar is in there because it allows, it prevents the oxygen from dissolving all the way through the tube. All right. Does anybody have any questions so far before we go on and do a little experiment? If you have any questions, feel free to type them into the chat and we can talk about them. The one thing I do want to mention about these tubes, because we're going to use these in our experiment, the bacteria don't grow in these tubes exactly like those pictures we looked at. You will see anywhere that you get a heavy growth, you will see all of those cells clump together. And in the parts of the tube where you get less growth, the cells are not going to be all nicely suspended. They're going to clump together too. So often what you end up seeing are these strings hanging down into the lower parts of the tube. Those strings are just cells that have clumped to each other as they're growing. The agar does this. The agar prevents the cells from just, being all the way through the tube. So if you got a heavy growth up in the oxygen, you'd get this nice thick, like pack of cells up top. And then you'll see these strings hanging down if the organism can grow where there's little oxygen or no oxygen. All right. So here's our experiment. We're going to look at 4 different organisms and we're going to try to figure out which oxygen category they belong to based on the pattern that they grow in a thioglycolate tube. Remember, we can look at that pattern of growth and we can assign them to an oxygen category. So our first organism is called Clostridium Sporogenese. This one is a gram-positive rod. You can find this organism living in the intestinal tract. You can also find it out in the environment. It likes to live in soil and it likes to live in sandy sediment at the bottom of the ocean. This is what it looks like on a slide. This is a smear of Clostridium. You can see it's a gram-positive rod. Remember, Clostridium is one of the 2 genera of bacteria that can form spores too. Second, we have an organism called Enterococcus Fecalis. This is also a gram-positive organism, but this one is a coccus. In fact, some references call this cell a coccobacillus. I don't know if you remember, but when we were talking about cell shape, we said that there is a category of shape that's halfway between a coccus and a rod. And this is an organism that kind of fits that. So if you look at some of these cells, they look quite spherical. They look like cocci. Like here you can see two cells side by side and they look nice and round. They look like cocci. But if you take a look at this chain here, or if you look at these two cells right here, some of the cells look a little more elongated. They're not quite rods, but they're a little bit longer than we might expect a coccus to be. So some references, not all, but some reference texts will call Enterococcus a coccobacillus. Just like Clostridium, this one lives in the intestinal tract, and it can be found in other parts of the body. Here's our friend E. coli, Escherichia coli. This one is a gram-negative rod, and it lives in the gastrointestinal tract. You can see them magnified a little bit here. 1, 2, 3 cells. Those are rods. And last, we have an organism with a terrible name to pronounce. It's called Alkalogenese Fecalis. This one is a gram-negative rod, and it lives both in the intestinal tract and out in the environment, just like Clostridium does. So we have 2 gram-positive organisms, and we have 2 gram-negative organisms, and we want to know what their oxygen requirements are. So before I did this experiment, I verified that all of the cultures were alive. And the other thing I want to remind everybody of is that all of these organisms can be found in the gastrointestinal tract. So they are perfectly harmless most of the time. They're normal parts of our microbiome. But just like all bacteria, there are pathogenic strains of these organisms. So yes, you will find E. coli, you will find Enterococcus in the intestine normally, but there are some strains of these bacteria that are pathogenic. So we've got thioglycolate broth today. We've also got some nutrient agar plates because we want to put the cells onto plates so we can use the Brewers jar. We will make 2 nutrient agar plates for each organism. So one can go into the Brewers jar and one can stay out of the Brewers jar, in other words, and just go in the incubator. And the last thing we'll do is some catalase testing. So let's do a little bit of prediction before we begin looking at the results of this experiment. First thing to ask is, what do you expect? What do you expect to happen? What do we think, for example, will grow on a nutrient agar plate that's incubated in the presence of air, in the presence of oxygen? Well, obviously, anything that's an obligate aerobe is going to do fine on that plate. But that's not the only kind of organism. Facultative anaerobes will grow. They like oxygen. They prefer oxygen, so they're going to do fine. We'll even get aerotolerants and micro aerophiles to grow, but they're going to grow poorly. Remember, both of those categories prefer to be in low oxygen environments. So they'll grow on those nutrient agar plates, but they're not going to grow well. The colonies are going to be smaller than we would expect to see. What should grow on a nutrient agar plate in a brewer's jar where there's no oxygen? Obviously, the obligate anaerobe, because oxygen is toxic to them. But again, a facultative anaerobe is going to do fine in there too, because they don't require oxygen. They grow best in oxygen, but they don't require it. So they're going to be fine in that jar. Aerotolerant organisms will also grow in there, but they're going to grow poorly. Remember, aerotolerants grow best in low levels of oxygen, but they can grow without oxygen. What about Catalase Test? Well, obligate aerobes, facultative aerobes, and micro aerophiles are going to have a positive catalase test. These are organisms that are around oxygen. Obligates have to have oxygen. Micro aerophiles have to have low levels of oxygen. And facultatives prefer oxygen. So we would expect them to have oxygen around them. We would expect them to make hydrogen peroxide, and they would need catalase to break it down. Who should have a negative test? Well, the other two, obligate anaerobes and aerotolerants will have a negative test. We can refer back to this picture as we need to when we look at our results. So let's talk about what we got. When I looked at the plates, and I apologize, I don't have pictures of these. The pictures did not come out for these. They were blurry. But on the nutrient agar plates that were left in the incubator exposed to the air, the Clostridium Sporogenese, no growth. No growth. The Enterococcus Fecalis, it grew, but poorly. The colonies were really small. E. coli grew really well, no problem at all. And Alkalogenese Fecalis grew really well, no problem at all. Now, the nutrient agar plate that went into the Brewers jar, where there was no oxygen, the Clostridium Sporogenese grew great. Lots of good, normal-sized colonies on that plate. Enterococcus Fecalis grew great. Lots of good, normal-sized colonies. E. coli also grew, but it grew really poorly. And Alkalogenese, it didn't grow at all. Interesting results. I would say that we could already make a couple conclusions before we even look at the thioglycolate, couldn't we? Clostridium Sporogenese didn't grow at all when we put it on a nutrient agar plate and put it in the incubator. No colonies, none. But when we put it on a nutrient agar plate and we put that plate in a brewer's jar and took all the oxygen away, it grew great. What category do you think Clostridium belongs to? Just by those results. Cannot grow in room air. Can't do it. But put it in an environment where there's no oxygen and it grows great. What do you think? Obligate aerobe? Obligate anaerobe? Facultative? Aerotolerant? Micro aerophile? What do you think? This is one of those extreme results, right? No growth on one plate, plenty of growth on the other plate. Izzy's got it. Morgan's got it. Clostridium is an obligate anaerobe. Does not use oxygen to make ATP, and in fact finds oxygen toxic. Oxygen will kill it. How do you grow it? Well, you put it in an oxygen-free environment. If it gets exposed to oxygen, it's going to die. It's an obligate anaerobe. Now, how about alkalogenese? Alkalogenese did exactly the opposite. Alkalogenese grew great on the plate that was left in the air and didn't grow at all in the brewer's jar. What do you think for alkalogenese? Alkalogenese grew great on that nutrient agar plate that we just put in the incubator. But when we put it into a brewer's jar and took all the oxygen away, we got nothing. Which category does alkalogenese fall into? It's another sort of extreme example, right? Grows great when it has air around it. Cannot grow at all when you take the oxygen away. That's right. That's right, Izzy and Morgan and Kaylin. Good. Obligate aerobe. Requires oxygen. Needs oxygen to make its ATP. Very good. Now the other two, those are not as easy yet. We need some more results for the other two. We have some that, grow poor in one case and better in another case. So we're really going to need the Thioglycolate tubes for the other two. But before we look at those, let's make a prediction. Where in the Thioglycolate tube do you predict the Clostridium is going to grow? Oxic zone? Oxic zone and the rest of the tube? Bottom of the tube? Where do you think the Clostridium organism is going to grow in here? As an obligate anaerobe. Am I going to find it all the way through, or am I going to see a clump at the top and some strings? Am I going to see a clump at the bottom? What do you think we're going to see? It's an obligate anaerobe. What do you think? Oxygen is toxic to that organism. Izzy's got it. Bottom of the tube, way down at the bottom. Alkalogenese is going to be the opposite. Alkalogenese should be up at the top, right? We should only see alkalogenese in the oxic zone because that organism requires oxygen. It's got to have it. The only place you can find 20% oxygen is up at the top in the oxic zone, the top 20% of the tube. Okay? And we'll have to see what the other two do and see if that helps us put them in a category. All right. Here are our thioglycolate tubes. Here is the Clostridium tube. Now, this is a great example of how sometimes in the laboratory, things don't go the way you want them to, the way you would expect them to in the textbook. Do you see this little white clump right there? That's our organism. That little clump down at the bottom of the tube. That's all we got after 24 hours. I would have expected to see a big mat of material down in the bottom of this tube, but we only got a little clump. Listen, Clostridium is not easy to grow because oxygen is toxic to it. So it could be possible. It's possible that that organism just had trouble. Might have gotten exposed to a little bit of oxygen when it was being moved around. But we did get some growth, And we only got growth in the very bottom of the tube. Let's see how our guess went for alkalogenese. Here's the alkalogenese result. Take a look at that. See that mass of material? That's what I refer to as a mat. It's just a big clump of bacterial cells all growing and sticking together. And boy, oh boy, it is limited to the top of the tube. It's only in the oxic zone. This is our obligate aerobe, just like we predicted. All right, now let's look at the other two. This is Enterococcus Fecalis. Now, this is what I mean by strings. Can you see those? There's a mat in here. There's a big mat right there. So the best growth is right here. And then we can see fewer cells growing down, down, down throughout the rest of the tube, all the way to the bottom. These are cells, but they're all clumping together. And there are fewer cells making this string than there are at the top of the tube. So obviously, there's nothing at the very top. So the top half of the oxic zone, no growth. The bottom half of the oxic zone, lots of growth. In fact, the best growth. And then less growth, but still growth throughout the rest of the tube. Enterococcus. What do you think for Enterococcus? It's not growing all the way through the oxic zone, is it? It's only growing in the lower part of the oxic zone. It's growing best there, though. We also have growth through the rest of the tube, but to a lesser extent. What do you think? I'm going to pull the pictures back up so we can remind ourselves. Take a look at the pictures. Let me pull this one up because I think it's our best. You have to excuse my dog. She's barking at the delivery man. So here are our patterns. We've got an organism that's growing really well in the lower half of the oxic zone, but also less well through the rest of the tube. Where would you put Enterococcus Fecalis? Izzy's got it. Morgan's got it. Aerotolerant Anaerobe. Aerotolerant Anaerobe. Remember, an aerotolerant doesn't use the oxygen to make its ATP, but it tolerates it. In fact, it likes to grow in a little bit of oxygen. So that's why we get that nice mat in the lower part of the oxic zone. Aerotolerant. Good. Let's look at the last one. Scroll back down here. Here's our friend E. coli. Now you're going to see there are two tubes for E. coli. I didn't like either tube perfectly, so I chose two different tubes. These are tubes that students made for me. I want to show you the tube on the right first. This tube has heavy, heavy growth in the oxic zone. That's where it looks sort of cream colored. And then I think you can just see there's a string that's starting to grow right here. It's not a very big string though, is it? It's just faint, but there is a string growing down in the bottom. That's why I also like this tube, because this tube has much more evident strings coming down. So we've got an organism here with E. coli that grows very well throughout the oxic zone and can grow less well throughout the rest of the tube. We have two categories left. We have facultative anaerobe, and we have micro aerophile. And our organism, E. coli, is growing very nicely in the oxic zone, very nicely up at the top, and has strings hanging down. It grows all the way through the oxic zone and then has strings hanging. Best growth is in the oxic zone. You're doing great today, Izzy. You're doing great. You got it again. Who else? Who else can get it? What kind of organism that we talked about grows really well in the presence of oxygen, prefers to be in oxygen, because if it has a choice, it's going to do aerobic cellular respiration. But if there's no oxygen around, that's okay. It's going to survive, because it can also do anaerobic cellular respiration. It just switches pathways. That's right. Good. Good job, Kaylin. Good job, Morgan. Facultative. Facultative anaerobes grow like this. E. coli is a classic example of a facultative anaerobe. E. coli grows in your intestine, right? Normally. Doesn't hurt you. It's part of your microbiome. It's down in an environment where it's getting oxygen in your intestine, but it's not like it's out in the world. But E. coli can survive in water. It can survive in soil. It can survive in all kinds of environments because it's capable of switching pathways like that. Now, the one organism that we didn't get to see is a micro aerophile. We didn't have an organism today that we could grow that would show us the micro aerophile pattern. Remember, when you have a micro aerophile, that's an organism that requires oxygen, but it requires low levels of it. That's the micro name. Aerophile means it loves air. It loves oxygen, but it wants a low concentration of oxygen. So if we had had a micro aerophile, we would have seen growth only in the lower part of the oxic zone. Micro aerophiles are hard to grow. They're just hard to grow. And there aren't that many of them compared to something like a facultative anaerobe. There are lots of facultatives out in the world. Good job. All right. So these last couple of slides just have the conclusions that you've just told me. Clostridium Sporogenese, we said, was an obligate anaerobe. Doesn't grow in room air, grows great in a brewer's jar, and grows deep down in the anoxic zone of the thioglycolate tube. Alkalogenese Fecalis, obligate aerobe, grows great in room air, no growth in the brewer's jar, grows only in the oxic zone of the thioglycolate tube. Enterococcus grows in room air, also grows in the brewer's jar, but not as well. Grows in the lower oxic zone. That was really the key here, as well as the lower anoxic regions of the tube. That was our Aerotolerant Anaerobe. And E. coli was our last one, grew in room air, grew in the Brewers Jar, but not as well. And it grew all the way through the Thioglycolate tube, although the heaviest growth was in the oxic zone. E. coli is a facultative anaerobe. All right. So when we have an unknown microbe, when we get a sample in from the environment or from a patient, and we have to identify a bacterium, one of the things we can do is figure out what its oxygen category is. And we can do that by inoculating thioglycolate tubes. And the pattern in a thioglycolate tube will tell us what category it belongs in. We can also grow those organisms in the air or in a brewer's jar and sort of confirm their status. We can combine both of those methods and we can really narrow down what category they belong to. Each different species of bacteria belongs to one of those categories. So it's a defining characteristic for a microbe. All right. Any questions about what we've covered today? I know we're over time now. I want to let you go. Any questions? All right. When we get together next time, what we're going to be talking about is disinfectants. We're going to talk about categories of disinfectants, big families of disinfectants, chemicals that are used to control the growth of microbes. We're going to talk about what the advantages are of each category and what the disadvantages are, what they're effective against and what they're ineffective against. So that will be what's happening when we get together on Thursday. Take a look at your pathogen instruction sheet. Let me know if you have any questions. And I will see you next time. I hope you all have a good evening.